Yes, we are. All right. Cell division. Huh? All right. Here's the question. Okay? The question is, how does an organism go from this small little baby or a small little plant shoot or something to this? A full-grown adult or a full-grown oak tree? Okay? We know from our lab on Friday that these cells that we're talking about are actually very, very tiny, right? Super duper small. So we know that multiple cells combine to form what? Tissues. Multiple tissues combine to form?
Organs.
Multiple organs combine to form? Systems. And multiple systems combine to form? Organisms. Okay? So we know that, but the question then is, how does one tiny cell start off small and then get to become a bigger creature? Okay? The answer is through cell division. Here's where the notes start. In order for multiple cells to form, each cell must reproduce. The first blank. In order for multiple cells to form, each cell must reproduce. Cell division is when existing cells divide to form new cells. Okay? It's exactly what it sounds like. Cell division is when existing cells divide to form new cells. When a cell divides, it forms an exact replica of itself, which means it is a picture-perfect copy. They are exactly the same. Okay? If a nerve cell is going to divide and reproduce to form another one, it's not going to reproduce a skin cell. Okay? A muscle cell will not multiply into a bone cell. Okay? A red blood cell will not multiply into a white blood cell. All right? When a cell divides, it forms the exact same copy of itself. That goes right along with our cell theory, right? Part 3 of the cell theory says that all cells come from other living cells, or pre-existing cells, is the same thing. Other living cells is what we have in our notes. Okay? So these cells divide through a process, and that's what we're going to look at today. Okay? Micah. About millions of cells in your body are reproducing every second, but at the same time, about millions of them are dying as well. They die and reproduce all the time. Matthew?
So, what happens if a blood cell split and reproduce, and would it reproduce another blood cell? Would it reproduce like a red blood cell or a white blood cell?
Depends on whatever kind of cell is reproducing, it creates an exact copy of itself.
Oh, because you said a blood cell would divide to form a white blood cell.
Right. It would divide to form another red blood cell. The white blood cell would divide to form another white blood cell. Sorry, a red blood cell. Okay? All right, so why do cells divide? There's 2 reasons. Number 1, to allow an organism to grow larger. That's the first reason, to allow an organism to grow larger. Okay? Think about our muscles, right? Our muscles, made of muscle cells, grow and get larger as you work them out because you're helping your muscles to create more of themselves. Okay? Now, some of that relies on DNA. Okay, no matter how much I work out, I will never look like Dwayne The Rock Johnson. Okay, which is totally fine by me. But part of that is also how much you're working it. Okay? Also, it allows organisms to grow larger, even bone size, right? The size of my bones now is not the same as the size of my bones when I was a baby. Promise. Okay? As you grow, your cells multiply to help you grow larger. The second reason that cells divide is also to help organisms replace injured cells. When you get a cut or an injury or you get hurt in some way, the reason why it does not stay like that forever is because your cells divide to form new cells to replace the ones that you hurt. Okay? Now, scarring happens when, number 1, the injury is really bad or really a deep cut, or if you had a scab that you picked off over and over and over again. Okay? It's just basically creating a bigger divot, and your body can't quite recover from that. Or it maybe it will take a long time for it to recover. Okay? But no matter what, the cut will not stay there all the time. Eventually, your cells will create more of them to replace the ones that you hurt. Yes. Okay, make it fast.
So, when a cell divides, it makes an exact copy.
Mhm.
So, they're two of the exact same cells, but then if one of them dies, the other one dies the exact same.
Good question. Probably not. It would depend on why that cell died. Let's say that maybe in the process of cell division, that one cell didn't get enough nutrients to survive, then it would die, but the old one should be fine. Once they split and divide, they're not like connected anymore. They're just two individual cells. Whatever happens to this one may not happen to this one. Okay?
How come sometimes when I get a contact, you can only like see it.
You can only see it. Oh, you can't see it at first. You can see it the next day. Because sometimes you can't see it until the scab forms. Because the scab is usually darker than the cut. So because the scab is darker, that's why you see it. Okay? All right. No more questions right now. We'll get to them just a little bit. Okay? When a cell copies itself or replicates itself, the new cell is called the daughter cell. I'll help you. Okay. Now, this does not mean that the new cells are girls. It doesn't mean that they're not boys. Okay? It just means that they came from the parent cell or the original. Someone in the first class, Vivian, actually pointed out that maybe they're called daughter cells because these cells can now go on and reproduce. Okay? And have more. Okay? So that may be why. I'm not really sure, but it's a great thought. So in either case, the parent cell, once it splits, it creates two new daughter cells. Okay? The daughter cells are what we call the new cells from division. All right. So where does all this take place? It begins in the nucleus. Okay? Which makes sense. We know by now that the nucleus is like the headquarters of the cell or headquarters of the city, the mayor's office. It's in charge. So for an important process like dividing, okay, everything should start at the headquarters office. Yes.
I don't really understand wait, isn't the nucleus a daughter cell?
You'll see. We're going to go through all 5 phases. It's a whole process. Okay, so it begins in the nucleus. Before a cell can divide, the DNA in the nucleus copies itself. Okay? So, the DNA, we know, is the instructions for the entire cell. So, if a new cell is going to be formed, it needs a copy of those instructions, right? It needs a set of DNA. So, if we look at a cell that's about to divide, let's say this is the nucleus, we know that the DNA is kind of all stringy in there. Okay? Before it divides, it makes a whole nother step. So now this nucleus is extra full because it has 2 copies of DNA, not just one. Okay? That's the first step. The DNA copies itself. When this cell divides into 2 new cells, this doubled nuclear material splits in half so that each daughter cell is the same as that of the parent cell. So this doubled nuclear material, one set of DNA will go to one of the new daughter cells. The other set of DNA goes to the other daughter cell. But both have one exact full copy. Here's what doesn't happen. Let's pretend I'm going to share my book. Okay? Edgar and I are going to share a book. If I give him half my book, does he have a full copy of the book?
No.
Do I have a full copy of the book?
No.
No. If I want to give him a full copy of the book, I have to take my book, make a whole separate copy of it, and then give him a full one, right? Same thing happens with the DNA. When it splits, it doesn't get half and half. It has already made a whole new copy so that when it splits, each one has a full set. Does that make sense? Okay. The DNA is an exact match. And that's the next part of your notes. The DNA is an exact match. The dividing of nuclear material, Okay, this process of taking the DNA in the nucleus and dividing into two is called mitosis. Say mitosis.
Mitosis.
Good. Now, there are 5 different phases to mitosis, the step-by-step process. We're going to look at each one of them, okay? But for right now, you just need to know that it's called mitosis. In the very last stage of cell division, okay, so mitosis is done, the nucleus is divided. The last part of the cell dividing is the cytoplasm will split in half. Okay? The cytoplasm divides as well, making 2 daughter cells where there used to be 1. Okay, you can kind of see the cytoplasm pinching right there. Okay? So, cell division is the process of a cell dividing itself. The part of that where the nucleus divides itself, that is called mitosis. It's a part of cell division. Does that make sense? Okay. Caleb, question? Brian?
So what if you're a prokaryote cell and you're in cellular organism?
Mm-hmm.
Do you split and divide? Because then there'd be two cells.
They have a process where they also split their DNA, like copy their DNA and split it, but it's not mitosis. This process that we're going to study today only applies to eukaryote cells because it happens within the nucleus. In a prokaryote, they don't have the nucleus, so this doesn't happen. They have a different replicating process.
But how does skin have 2 cells and organism, right? So what do you have to do?
The new one is just a brand new organism. It's like having a baby. It has a brand new thing, living thing, because that one cell is the living thing. Good question. Aiden? So one of the daughter cells used to be the parent cell. It's just once it reproduces, it now has this, now it's called a daughter cell. Okay? All right, one more, then we're going to move on. Wyatt. Once it divides and splits, each of the 2 new nucleuses will have all the instructions it needs to make new organelles. So I'm assuming, I don't know this for sure, but I think from what I've studied, which is not much on this part, I think the organelles that have that kind of are still there, they basically just split. Okay, so maybe some of them went over here, some went over here. Whatever they're missing, they then create once the new cell is created. That could be very wrong, but that's what I think I know. Okay, hold on. I'm going to get to more questions in a little bit, okay? I know it's a lot. All right. Before we get into it, the DNA during cell division. Now, we know that typically DNA is all stringy, right? It's spread out, this tangled form of DNA, chromatin, right? It looks like spaghetti. Before a cell divides, the first thing that has to happen is the DNA shortens itself. It doesn't like cut anything off, but instead of being all long and strung out, it scrunches itself up. It kind of coils up, okay? When it coils up, it coils into this X shape called a chromosome. Remember our term chromosome from the vocab? Maybe, kind of, sort of? Okay. Chromosome is the shape the DNA makes when it's kind of coiled up, okay? Now, remember, I showed you before how we had 2 sets of DNA, right? I showed it with blue and red. When the DNA scrunches up before it divides, it forms this X shape, or this chromosome, okay? With one side having one copy of the DNA, and the other side having the other copy of the DNA. Okay? And there would be multiple chromosomes throughout the cell before it divides. Does that make sense? Okay? So the DNA scrunches up into these kind of more compact forms. They're smaller. It's not all spread out and stringy and messed up. It's all compact into little X's. Okay? Does that make sense? Does that make sense? Okay. Perfect. Okay, so stages of mitosis. I'm going to play this video for you first, okay? And then this video kind of goes through all the phases at one time, all right? And then I will show you, we're going to go through and do the notes for them, okay? Leave the lights on though because it's kind of dark outside. So, when we play this, it's going to kind of go through all the phases, like I said, and then at the end, we'll do notes about each one specifically.
Few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now...
Okay, so like in my example, this would be like the red copy or this would be the blue copy. Okay, so here's one set of the DNA. This is the copy that they're making. Okay?
Consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical
So it's
fibers.
scrunching up.
Outside the nucleus.
Okay, remember the churro-looking things from the animal cell? Okay, here they are. Okay, these are the centrioles. And the video is going to show you in just a second. The centrioles become important during mitosis because they begin to separate from each other. And as they do, they release these strings right here. The strings get longer and longer, and that is what the chromosomes will connect to. Okay?
Centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids.
Okay, so that's the DNA all scrunched up into a chromosome. Okay?
The chromosomes continue to condense. The nuclear envelope breaks up.
There's lots of them.
The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid.
So the chromosomes kind of attach to those strings, basically, and now you're going to see like some tug of war, basically. The chromosomes kind of wiggle back and forth until they calm down, and eventually they all line up in the center like this. Okay?
The chromosomes are bound by spindle fibers from opposite poles. They move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate.
Kind of line up in a straight line.
The sister chromatids are released from each other, each becoming a full-fledged chromosome.
Okay, so remember when I told you that one side of the chromosome was like the blue, one side like the red? Okay, when those split, now all the red will go to this side, all the blue will go to this side. And each one will have one full set of DNA. One blue, one red. You see what I'm saying? Okay, so anaphase is where the chromosomes split and begin to pull apart.
They are moved toward opposite poles of the cell.
So here's the blue, here's the red.
To them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart.
This is the last phase, telophase.
Once the chromosomes arrive at their destination, they become less condensed.
Okay, so once they get over here, okay, let's say pretend these are the red, these are the blue. All the chromosomes kind of spread back out into that stringy form of DNA. Okay, so instead of being all tightly bound together, now they kind of relax and spread back out. Okay, a new nucleus will form on both sides. Okay, you'll have two brand new nucleuses. In the middle part, can you see how the cytoplasm is kind of pinching like this? Okay, the cytoplasm will continue pinching until it splits all the way in half, and then we'll have the brand new cells. Okay, you'll see the nucleus will form here, and eventually this will split all the way. Okay.
two new nuclear envelopes form, completing mitosis,
There's a
the
nucleus and a
division of
nucleus.
one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming
There it
two
goes.
separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Okay, so that is kind of the overview process. Now we're going to slow it down and look at each phase in particular. Okay? That's a quick question. Okay.
So, so they divide in like, it takes them seconds to divide. Like, so it doesn't take as long as 3 minutes.
No. That just slows it down so you can see each part. Micah? Good question. No, they don't get smaller because that DNA has doubled itself, right? So, again, it's like the book example. I'm not giving him a smaller version of my book. I'm giving him an exact copy of it. Now, right as they split, they may be a little bit smaller than normal, but as it splits, it will very quickly grow back into a normal size cell. Riley?
Could you actually, like, if you're on the could you actually do it, like,
If you were examining live cells, I guess you could. But like in the lab, I think we had some slides where you could see like the cell or the organism was killed basically right during cell division. You can see some of them like about to split. Okay, hold on. Let me get to these and then we'll do more questions at the end. Okay, so first phase is interphase. Okay, this is when each chromosome makes an exact copy of itself. So the cell goes from looking like this to this. What's different here than in here? Well, the nucleus is way more full over here, right? It's created a whole nother copy of DNA. Okay, that's interphase. Second phase is prophase. Prophase, P-R-O, phase. After the DNA has made a copy of itself in interphase, now that tons of DNA has to shorten up and get a lot easier to move around, right? So prophase is when the tangled stringy form of DNA condenses into chromosomes. The genetic material shortens and thickens. The chromosome copies are held together at their centers, forming an X. Okay? Yes.
One question, but how does the organism make the organelles?
The instructions in the DNA has all of the instructions to make organelles. Okay? Third stage is metaphase, M-E-T-A phase, metaphase. This is the phase when the two copies of chromosomes line up in the center of the cell. So, the DNA was copied in interphase. In prophase, it formed the X shape. Now, in metaphase, after being kind of pulled back and forth between the centrioles, or centrosomes, can't remember. No. Yeah, centrioles. Yeah, centrioles. Okay? After being pulled back and forth, they eventually chill out and kind of settle into one straight line here. All right. Now, remember what I said. Technically, this side is one copy of the DNA. Okay. And then this side is the other copy of the DNA. Okay. So when it splits down the middle, all of this will form one set of DNA. And all of this will form one set of DNA. This will pull to this side. The blue will pull to that side. Does that make sense? Okay. So anaphase is when the chromosomes split. Or no, yes, anaphase. Metaphase, they line up. That's the third phase. Fourth phase is anaphase. A-N-A phase. The fourth phase is anaphase. This is the phase when the copies separate. One set of if I made this picture, I would put all blue on one side, all red on the other side. I think this is confusing. But anyway, They split down the middle. One set of chromosomes is pulled to one side of the cell. The other is pulled to the opposite side of the cell. You can also see in Anaphase, especially in this picture, can you see the bubbles on the outside beginning to pinch a little bit? Okay, almost like you have a rubber band around it and it's making it a little bit smaller. Okay, kind of like a balloon. All right, so that's Anaphase. The chromosomes split and begin to separate. In Telophase, this is actually spelled wrong. It should be Telophase. T-E-L-O phase. It's phase number 5. T-E-L-O phase. Intelliphase is the final stage of cell division. During this stage, the cytoplasm pinches in at the center of the cell, dividing the cell in half. 2 new daughter cells are formed. The daughter cells are identical to the parent cell. So once the chromosomes split, these guys will begin to relax and spread back out into a regular stringy DNA. Okay? A nucleus will form on this side around the DNA, and a nucleus will form on this side around the DNA. Okay? The cytoplasm pinches all the way until it eventually splits it in half. Then you have your 2 new daughter cells. Does that make sense? Ask you a question.
So does it form kind of like if you have this cell right here, does it form like this?
Sort of. So like if this is the cell, if this is the cell, I kind of think about it like maybe it gets a little bigger maybe, and then it just whoop. Or like if this is the cell, it just kind of and splits into 2. Except the new ones are they're not actually smaller. Eventually they will become bigger just like this.
And how many cells if there's one right here and it splits in half, then wouldn't it be smaller?
It may be smaller for a second, but once they become new cells, they have all the DNA, right? And they have all the instructions to create everything they need in the cell.
Oh.
So it would eventually then grow to a full-size normal cell.
So, what happens with the organs in the cell?
The organelles. Now, did I already answer this? I can't remember.
Yes.
I think Wyatt may have asked this, but this is what I know. I'm not 100% sure if it's correct, but I think the organelles just kind of hang out around the outside here while this is taking place. And then as the cell splits, they kind of just go whichever way they go. And whatever they're missing, the DNA will then build in the new cell. Now, again, that's my assumption. I'm not 100% positive. Well, remember, in the nucleolus, that will create the ribosome for the brickyard. Okay? All right, we'll get to more questions in just a second. But right now, go to the bottom of your notes. It says list the phases in order. We're going to list them as well as draw the picture. So I'm going to use Brian as my all right. Now, if you have 2 colors, I would recommend using 2 different colors. If you're using, you can even use pencil and red pen if you want. But I think it will just help you to see 2 different kinds. Okay? So, list the phases in order. All right. So, the order of the phases goes like this. I, P, M, A, T. IP mat or IP mat. That's the way to remember it. Okay? The I stands for what, Jacob? Interphase. Very good. So we're going to write interphase. Then we're going to draw a picture. Jacob, what happens in interphase? The DNA makes a copy of itself. Okay? So I have my nucleus here. And I'm going to draw 2 sets of DNA. I'm going to make one green and one red. Two copies of DNA. It's a very, very full nucleus. All right, what does the P stand for? We have interphase, then Chloe? Prophase. Very good. Chloe, what happens in prophase? Okay, the nucleus does that, or the DNA in the nucleus? Very good. So I'm going to draw the nucleus a little bigger so I can draw my pictures. But the DNA shortens and thickens itself. Instead of being all strung out like that, it kind of coils up into these X shapes. Now they're not lined up yet, so don't draw them in a line. But I'll have one color making one side of the X, the other color making the other side. Okay? Next we have the M. What does the M stand for? Luke? Metaphase. All right, Luke, what happens in metaphase? Very good. So now we're going to draw our chromosomes again, but this time they're lined up in the middle. I only have space for 3. That's okay. So the chromosomes line up in the middle. Oh, it's a perfect line. All right, what does the A stand for, Connor? Anaphase. I'm going to draw mine over here because it gets a little stretched. All right, in anaphase, you kind of have to draw almost like a peanut shape, like in our mitochondria, because the cell is beginning to split, but it's not splitting just yet. Okay? In anaphase, that's where they divide. All right? So we have the green chromosomes or chromatids, whatever they are, come over to this side, and they're eventually going to kind of detangle themselves and kind of spread out into what we're more familiar with DNA, this stringy kind of spaghetti looking stuff. And then the red copies will go to the other side. And then what does the T stand for? Brooklyn? Telophase. Good. So T-E-L-O phase. All right. And then in telophase, we have the two cells are basically split. So I'm just going to draw a little connecting line there in the middle. And this time we know that the DNA is kind of back strung out. The new nucleus has formed. We're just waiting for the cytoplasm to finish kind of pinching off there. Okay? Now, once you finish that, I want you to take out your study guide because there's one part we need to finish up on that study guide. Keep in mind, on your test, you will not have to list and describe each stage. That's what it says on the study guide right here. Give a brief description of each stage. You will not have to do this on the test. Instead, it will be picture labeling. I will show you one of these pictures. For example, maybe I'll give you, this one, that one. And you have to figure out, okay, what's happening in that picture and what phase is it representing. Does that make sense? Take your phone with you. All right, any questions about how we're going to do that? Are you going to have to give me this? No. I will give you the pictures of each phase. You tell me which phase it's showing. Okay? All right.